Hello learners, this is the lecture on genetic code. I'll be talking about important characteristic of genetic code with a special emphasis on wobbling hypothesis and exception to the rule of universality. Now, first of all, let's see what is central dogma of genetics. Central dogma of genetics is where we talk about the synthesis of mRNA from the DNA and then formation of protein from mRNA. So the transcription of the gene to produce your mRNA and then translation of mRNA to produce your protein is called central dogma of genetics. Now, when the translation is taking place from mRNA to the protein, when the nucleotide sequences are read for the sequence of amino acid, there are certain set of rules which are being followed. The connection between the mRNA and the protein is the process of genetic code. So let's see what is genetic code. What all rules are there for the genetic code which transfers the information from mRNA to the protein where sequence of nucleotides are reflected into the sequence of amino acids. We have many characteristics of genetic code. I'll be discussing them one by one. Number one characteristic of genetic code is that one codon, which is the basic unit of an amino acid, is actually made up of 3 nucleotides together. You should know that we have altogether 4 variety of nucleotides on mRNA. 4 variety of nucleotides in mRNA. Now 3 nucleotides. There are 4 variety of nucleotide. 3 random nucleotides, they code for 1 amino acid. It is not 2 nucleotides which code for 1 amino acid. It is not 1 nucleotide which codes for 1 amino acid. Suppose if 1 nucleotide is coding for 1 amino acid, then we will have only 4 amino acid being coded by 4 different nucleotides. This is not the case. Similarly, suppose if 2 nucleotides together are coding for 1 amino acid, the differential combinations of 4 different nucleotides in a pair results in 4 into 4, that is 16 combinations. So that will be coding for only 16 amino acids, which again is not the case. So the rule says that we have 3 nucleotides which code for single amino acid. These are nucleotide, this is amino acid. So 4, 4, 4 results in 64 codons. So we have possible 64 codons, out of which 3 act as a stop codon and 61 codon code for amino acid, 20 amino acids. We have 20 amino acids which are coded by 61 coding codons. 3 codons are stop codon. So the codon is made up of 3 nucleotides. That is the basic rule of genetic code. And combinations which are possible when there are 4 variety of nucleotides and we have 3 in 1 codon, the possibility of variation in the codon is 64. And that is the reason why we have 64 codons. And out of 64 codons, we have 3 codons which act as a stop codon. They are UAA, UAG and UGA. So they do not code for any amino acid, and remaining 61 codon, they code for 20 amino acid. So this is the first rule of genetic code, where you should know that codon is made up of 3 nucleotides together. Now codons are non-overlapping. Now let me explain you this point. Suppose this is mRNA and this is 5' end, this is 3' end, and we have nucleotides on mRNA like this. These are nucleotides A, C, C, A, G, U, C. This kind of nucleotides are arranged on the mRNA. And when we are talking about reading of codons, please understand this is one codon, this is another codon, this is third codon, and they are being read in triplet fashion in a continuous manner without any overlapping, without any punctuation in between them. All nucleotides are being read. See, there is no gap. This is called no punctuation. It's a continuous reading frame. And we have 3 nucleotides which are read in this manner where there is no overlapping. So this explains the non-overlapping concept and no gap concept between the genetic codons. There is no overlapping. Overlapping means this ACC is one codon, and when the next codon is decided, the C is taken into consideration probably, and next 3 nucleotides are being read. Then maybe that A and G is taken into next codon reading and the next codon is decided. This is not the case. When we are talking about genetic code, there is no overlapping of the nucleotides in the codon. In other words, none of the nucleotides are read twice in genetic code. So this explains the phenomena of non-overlapping and continuous reading. Continuous reading also have explained you that there is no gap, there is no punctuation, comma-less it is. Means none of the nucleotides are missing in the reading. All nucleotides are being read in a fashion of 3. So this is non-overlapping and continuous reading of the nucleotides in a codon. Now they show the phenomena of degeneracy. Now I'm going to explain you the next point, that is degeneracy. They say genetic code, they degenerate. What is degeneracy? Can you define degeneracy? How to define the degeneracy of genetic code? In simple line, the degeneracy can be defined as multiple codons for the same amino acid. Degeneracy is multiple codons for the same amino acid. You have just seen that there are 61 coding codons. 3 are non-stop codons that I'm not considering here because they are not coding for any amino acid. So we have 61 coding codons which code for 20 amino acid. So this figure clearly tells that one particular amino acid is coded by more than one codon. I repeat again, more than one codon, they code for single amino acid. This phenomena is called degeneracy. So multiple codon for coding for the same amino acid is the degeneracy. For example, I have given you some illustrative examples here. Like if you talk about the proline amino acid, which is actually the amino acid, it's coded by CCU and CCC codons. Alanine has got so many codons: GCU, GCC, GCA, GCG. Leucine has got 6 codons: UAA, UUG, CUU, CUG, CUC, CUA. So multiple codons coding for the same amino acid. This is the degeneracy. But how this degeneracy is able to code for same amino acid? How different codons are able to code for same amino acid? This is a question which is very, very important question. How different codons are being read for the same amino acid? So this was explained by the Wobble hypothesis, which was given by Crick. So I'm going to next explain you wobbling, which is explaining the phenomena of degeneracy. Why wobbling is so important? Because it is explaining it explains the phenomena of degeneracy. It explains how different codons are being read for same amino acid. This I'll be explaining you shortly. So I just wanted to tell that we are discussing the characteristic of genetic code, where we have seen 3 nucleotides are making a codon, and codon reading is non-overlapping, and codons are read without any punctuation mark, without any comma. And they show the phenomena of degeneracy that I'm dealing in detail, where the wobbling I'm going to explain you, which is explaining the phenomena of degeneracy where multiple codons are coding for the same amino acid. What is wobbling? Wobbling was proposed by Crick, and he said that the third nucleotide of the codon, see this green color line is the mRNA and this is the third nucleotide of the codon. You should know that matching of codon with the anticodon of transfer RNA is antiparallel. You can see this is 5' to 3' end. This is being identified in 3 to 5 directions. So there is anti-parallel matching of codon and anticodon, and Crick said that this is the point which I have highlighted, is the point where wobbling is seen. Wobbling means Crick pairing. Watson-Crick pairing rule is not strictly followed. What is Watson-Crick pairing rule? That A will pair with U, C will pair with G. That is pairing rule. See, at first two nucleotides in the mRNA, the pairing rule is very well followed. But Crick said that this is the wobbling nucleotide. This is the wobbling nucleotide. That is the third nucleotide of the codon and the first nucleotide of anticodon when they are being read in 5 to 3 direction. This point, the wobbling is seen. Wobbling is non-Crick pairing rule. Not necessarily they'll follow a strict pairing rule. So the difference in the codon at third nucleotide can be masked. This is an example given in this picture where you can see both are codons for leucine, CUC and CUU. Both are being read for leucine carrying transfer RNA which are having anticodon GAG. How this anticodon GAG is able to read two different codon via wobbling at this point. You see the GMP at 5' end of anticodon of transfer RNA can pair with CMP at 3' end of codon of mRNA. Similarly, GMP at the 5' end of the transfer RNA can pair with UMP at the 3' end of the codon on mRNA. So this is called wobbling, where different nucleotides can be mashed at this particular position. This is the point. As a rule, the first two nucleotides are strictly pairing, but there may be wobbling at third nucleotide of the codon and the first nucleotide of anticodon when they are being read in 5 to 3 direction. This was proposed by Crick, and that could explain the phenomena of degeneracy, that multiple codons coding for the same amino acid. In a nutshell, the wobbling can be explained in this table. What is this table telling? The codon at 3' end of mRNA, suppose if it is AMP, then it can match with UMP or IMP. IMP is adenosine monophosphate, which is at times present in transfer RNA anticodon loop. So AMP at mRNA 3' end can match with either UMP or can match with IMP at anticodon loop at 5' end. And similarly, CMP at mRNA 3' end can match with GMP, which is as per rule, or it can match with even IMP at 5' end of tRNA anticodon. Similarly, GMP can match with CMP, which is as per rule, but GMP at 3' end of mRNA can match with UMP. UMP can match with AMP, GMP, IMP. So this table was proposed by Crick after many years of research, and he said that this is how the wobbling can take place, where 3' end nucleotide on the codon can show the wobbling at 5' end of the nucleotide of the anticodon in this fashion. This table can be represented in a different fashion where I can focus on first nucleotide of anticodon towards 5' end at the tRNA and third nucleotide of the codon on the mRNA. Third nucleotide. See, every time when we read the codon or anticodon, we have to read it 5' to 3'. So when I am telling you first nucleotide, means I am talking about first nucleotide towards 5' end in the anticodon loop. When I am telling you third nucleotide in the codon, I am talking about 3' end nucleotide of the codon. So that same table can be represented in other fashion like this, where the anticodon loop, if it is having the first nucleotide as UMP, it can match with AMP and GMP at codon. And if first nucleotide of anticodon at 5' end is having CMP, it can match with GMP. There is no wobbling seen here. Strict pairing rule. If AMP is there, it will pair only with UMP. No wobbling is seen. If GMP is there, it can pair with CMP and UMP as well. So IMP is there. It can pair with UMP, CMP, MP. So maximum wobbling is shown by inosine monophosphate, which can pair either with UMP or CMP or AMP correspondingly. So you can comparatively learn these two tables. These two tables are actually same. In the first table, I am focusing on the codon 3' end nucleotide, and in second table I'm focusing on the first nucleotide of anticodon loop of transfer RNA. This table is more or less same, like it is talking where all wobbling can be seen. You have seen that IMP shows the maximal wobbling. It can pair with AMP or CMP or UMP of the mRNA 3' end nucleotide. So this wobbling could explain how multiple codons are being read for same anticodon or same transfer RNA or same amino acid is being unloaded there. You have seen in the previous example when I said that leucine is having 6 codons. You can see that these 2 codons are having first 2 nucleotides same and only third one is different. So these 2 are identified by same transfer RNA. But if you focus on other 4 codons of the leucine, you can find the difference in them. The difference in them is on the third nucleotide. Over here also, third nucleotide. And you can see the first 2 nucleotides are CU, 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 CU. So the difference, if it is there in the first 2 nucleotides, then these codons are read by different transfer RNA. We have more than one transfer RNA for a particular amino acid. So if you're talking about these codons, and if you're talking about these codons, they are read by different transfer RNAs meant for leucine. But this transfer RNA can read any one of them because of wobbling, and this transfer RNA can read any one of these 2 because of wobbling. So wobbling is non-Crick pairing rule which is followed at a particular place, and I have told you this is the point where wobbling is seen, and this is called wobbling nucleotide. These are the wobbling nucleotide. Wobbling is not seen at first nucleotide pairing of the mRNA, right? So that wobbling could explain the phenomena of degeneracy, which is an important characteristic of genetic code, which explains how 61 codons, different codons, can be read for 20 different amino acids. More than one codon, how they are read for same amino acid, because transfer RNA tend to wobble at third base pairing. Unambiguous. What is unambiguous? Unambiguous means, if I write it here, if one codon is coding one amino acid, it is not going to code any other amino acid. Please understand. If one codon is dedicated for one amino acid, it's coding one amino acid, it's dedicated for it. It is not going to code any other amino acid. It's a different matter altogether that a particular amino acids are being coded by more than one different codons. But one codon is very specific for one amino acid. Like for example, if you see, if leucine is coded by UA, this UA is not going to code any other amino acid. CCU is coding proline, the CCU is not going to code any other amino acid. So this is the unambiguity. So try to understand the difference between unambiguity and degeneracy. Degeneracy, where multiple codons are coding the same amino acid. And what is unambiguity? That one codon, if it is coding one amino acid, that codon is dedicated for that amino acid. That's not going to code any other amino acid. That is called unambiguity. Now coming to the rule of universality. Previously, they used to say that genetic code is universal. Whatever the species, whatever the compartment of the cell, if one genetic code is there, that's going to code for one specific amino acid. But now we see that genetic code is not universal, it is nearly universal. So we have exception to the rule of universality in current date. The best exception to the rule of universality is shown by the mitochondrial DNA mRNA, where certain genetic codons are being read differently compared to cytosolic mRNA, which is coming from nucleus DNA. Now, if you focus on this table is talking to you about the exception to the rule of universality of genetic code. Like, for example, if you see, the first column is talking to you about those codons which are showing the exception to the rule of universality. Second column is talking to you about the cytoplasmic mRNA, how that codon is being read there. Like, for example, Yuga is read as a stop codon in cytoplasmic mRNA. Cytoplasmic mRNA represents the nuclear DNA. So Yuga is acting as a stop codon there in that genome. And AGA is codon for arginine, AGG for arginine, AUA for isoleucine. But let's see how these codons are being read in the mitochondrial mRNA. Mitochondria is also having its own genome and it's having its own mRNA. There the rule of universality is not exactly followed. There are exceptions, like for example, Yuga is read as a tryptophan coding codon in the mitochondrial mRNA, not as a stop codon. AGA and AGG, which is coding for arginine in the nuclear genome, it's acting as a stop codon in the mitochondrial genome. AUA, which is acting as a codon for isoleucine, this AUA is coding for starting methionine in the mitochondrial mRNA. So this is the rule of exception to the rule of universality, which you should know. So genetic code today's date is nearly universal. It is not totally universal. All right. So this brief discussion was on, I summarized everything once more. And in this, I will somehow fit this table also in my discussion. Now, let's see what we have seen in this short lecture. I was trying to explain you the genetic code, where the certain rules which the genetic code is following. I wanted to explain you. Now, in that context, first of all, I told you what is central dogma of genetics. Central dogma of genetics is nothing where the information from the DNA is transferred to the messenger RNA, and then the information of messenger RNA is being transferred to the protein. So when we talk about the transfer of information from mRNA to the protein, we understand that we are reading the nucleotides on mRNA and we are converting them into the amino acid sequence. So for this process of translation of mRNA to the protein, certain genetic codes are there. They are the codes which transfer the information from nucleotide to the sequence of amino acid. So let's see what is that characteristic. I explained you that they are codon. One particular codon which is meant for a particular amino acid is made up of not 1 or 2 or 4 nucleotides. It's made up of 3 different nucleotides. So differential combinations of 3 different nucleotides, which is of 4 variety, the possibility of getting 64 codons could be explained by this rule. And out of 64 codons, I said that 3 codons are a stop codon. They do not code for any amino acid, and remaining 61 codon code for 20 amino acid. This point itself tells you about the phenomenon of degeneracy, where multiple codons are coding the same amino acid, right? So these are the rules of degeneracy. Now, how the degeneracy could be explained, how different codons coding for same amino acid could be explained. In that context, I told you about the phenomena of wobbling, which was proposed by Crick, and he explained that at this particular position we have the wobbling, and this 2 table summarizes you the rule of wobbling, that if there are these codons at 3' end of mRNA, then how they can be matched with the 5' end anticodon nucleotide. Similarly, the other fashion, this table is represented in this, where anticodon is taken into account at 5' end, the first nucleotide of anticodon, how it can match with different codons. So there is a wobbling, there is a fumbling of the nucleotides at this particular position, which could explain that different nucleotides can be read by same transfer RNA, or if the nucleotides of the same amino acids I explained you are differing in the first nucleotides, then they are being read by different transfer RNA. So these things are very important and you should know about them. Now, I said that codons are not overlapping and they are read continuously. So when we are talking about the reading of codons, they are read in triplets and there is no overlapping between them. One nucleotide is never read twice, or none of the nucleotide is left behind from reading. So the reading is in a triplet fashion. There is no overlapping and there is no punctuation. There is no gap in the reading frame. So that explains the phenomena of continuous reading and no punctuation, non-overlapping. Then I explained you about the phenomena of unambiguity. Unambiguity means, what is unambiguity? Unambiguity, I have written here, that if one codon has coded for one amino acid, it is dedicated for that amino acid. It is not going to code any other amino acid. This is unambiguity. It is very important to differentiate these two characteristics. The number 1, degeneracy; number 2, unambiguity. Degeneracy means multiple codons for same amino acid. This is codon and this is amino acid. If we say degeneracy, this codon, and maybe this codon is coding this amino acid, this is degeneracy. And unambiguity means if this codon is coded this amino acid, it is dedicated for it. It is not going to code any other amino acid. This is unambiguity, right? Like if this codon is coding this amino acid, it's dedicated only for it. It is not going to code any other amino acid, right? So that is unambiguity. So it is important to differentiate between the phenomena of degeneracy and unambiguity. And when we talk about the rule of universality, now we say genetic code is not universal, it is nearly universal, because we have certain exceptions and mitochondrial genome is showing that exception. And this is the list of exceptions of rule of universality, where UGA is not read as a stop codon as in mRNA of cytosol. It is rather read as a tryptophan in the mitochondrial mRNA. AG and AGG, they act as a stop codon, which otherwise they are coding for arginine in the cytosolic mRNA. AUA is coding for isoleucine in cytosolic mRNA, which represents the nuclear genome, but AUA codes for a methionine in mitochondrial genome. So thank you very much, guys. So for more such videos, you can subscribe my channel. Thank you very much.